So I am Rishabh and I am a PhD scholar from IIT Kanpur, and today we'll be starting with the week 3 discussion of the NPTEL course, that is biochemistry. Okay, so starting with the first question. So you have given some statements regarding the polyacrylamide gel electrophoresis and isoelectric focusing. So a mixture of hypothetical amino acids A and B, their pKa1s and pK2s are given and can be separated into components using isoelectric focusing. So this is the first point. The second point is, in SDS space, the protein mobility is determined by both their charge and size. Okay, and the third point is, size exclusion chromatography is preferred for non-globular proteins. And the last one is, specific activity of an enzyme increases during purification because the activity of the enzyme increases on purification. Okay, so these are the statements, and you have to check which one is correct of all these statements and and choose the correct option. Okay, so please guess and let me know. So any answers? Okay, so someone said first, sorry, second. Any more answers? Okay, so the correct answer is option number 5. That means none of the options that have been given are correct. Okay, so let's see the statements first. So considering the pK ones, oh sorry, but first we'll calculate the isoelectric point, because in isoelectric focusing, the separation of the proteins are based on the isoelectric points. So likewise, Pi is calculated from P1's, sorry, pk1 plus pk2 divided by 2. So this is the average of both the pKs. And then for the first amino acid that we can see, the Pi is equal to by 2. So this equals around, so that means 6. And if we check for second amino acid, so this also comes out to be 6. So if the two proteins have the same Pi, that means we cannot separate them using isoelectric focusing. Okay, so this option goes wrong. Then in SGS space, the protein mobility is determined by both their charge and size. Okay, so because in SGS page, the proteins are not separated on the basis of their charge. Why? Because we are using SDS. That provides negative charge to the protein. Okay, so the whole charge of the protein, inherent charge of the protein, goes to only SGS that provides the negative charge. Okay, and only the proteins are separated based on their size. Okay, so the protein mobility depends mostly on their size rather than their charge. Okay, so this option also goes wrong. Then size exclusion chromatography is preferred for non-globular proteins. So instead of non-globular proteins, size exclusion chromatography is generally preferred for globular proteins. Okay, because these have a nearly spherical shape that can easily pass through the column through the pores of the column that we have been set for the size exclusion chromatography. Okay, so this option also is wrong. Then specific activity of an enzyme increases during purification because the activity of the enzyme increases on purification. So yes, this statement is correct. Okay, when we purify the enzyme, for example, we have the cell lysate, then we have certain steps of chromatography to separate or purify the enzyme. Okay, so in that case, what happens? The activity of the enzyme increases because the purity of the enzyme increases. Okay, so this option is correct. Although only D option is correct, that is not mentioned in any of the options. So option number 5 is correct for the given question. Okay, is it correct? Any doubt? Okay, so shall we move forward?
আমার এখান থেকে কি করে লিখতে হয় রে স্টেটমেন্টের?
Is there any doubt? Okay, if not, then we'll move on to the next question. So, wait a minute. Okay, so a biochemist purifies an enzyme generating the following purification table. So generally, when we purify an enzyme or a protein from a given cell lysate, then we develop a purification table. Okay, so this purification table gives us the amount of protein that we are extracting at a particular step and its activity. Okay, so that is calculated in units. So this is the first step, that is the crude extract, followed by salt precipitation and exchange chromatography, affinity chromatography, and gel filtration chromatography. And this is the amount of the protein that has been given. And this is the activity at that particular step after calculating the total protein in this. Okay. So now we have to check what is the most effective step in the particular process. Okay. So please let me know. Okay, someone said 2. Any more answers? 3. Okay. 3, 4. Okay, so let's start. So the correct answer is option number 2. That is the step number 4 is the most effective step. Okay, so how will we calculate that? So we will not be able to tell what is the most effective step from the data that is given directly. Okay, now we have to calculate the specific activity of the enzyme. Okay, so specific activity basically gives us the activity of the enzyme per total protein concentration of that of enzyme at that particular step. Okay, so this is the basic how to calculate the specific activity of the enzyme. So what is the major meaning of specific activity is that it gives us the enzyme activity at that particular step when we are counting on the total unit of the total protein that is present in that particular solution. Okay, so if we calculate the specific activity of the first step, then that comes out to be 50, right? Followed by the second step, it comes out to be 600. We'll just divide enzyme activity by the total protein concentration, total protein amount that we are getting. Okay, followed by the third step, this would come around 4000. Okay, now the fifth step, it would be around 15,000. Okay, now similar will be the for the fifth step. So as we can see, the specific activity jumped from the third step, that is from the ion exchange chromatography to the affinity chromatography, right? So here it's 15,000. So that means when the specific activity increased from 4,000 to 15,000, the enzyme purity increased. So here the most effective step would be the affinity chromatography, because here we are getting more number of enzyme molecules per unit volume that we are getting of the solution. Okay, that's why it is the most effective step of the whole procedure. Fine. So hence the correct answer would be option number 2, that is the most fourth step. Okay. So any doubts in this question? Hmm. Okay. So let's move forward. This is a similar type of question, but here, okay. So what happens? We have to check that from where the specific activity increased and it's reaching the maximum. Okay. If there would be some more specific activity of the fifth step, for example, if the specific activity jumped to 20,000, okay. So in that case, the most effective step will be the fifth one rather than the fourth one. But here the specific activity is same, that means the this has reached the maximum purification, maximum purity of the enzyme. So now it will not increase, that means it is saturating, right? So that's why this step, that is the affinity chromatography, will be the most effective step of the uh, full procedure. Okay, is it fine? Okay, so let's move on to the next question. So this is the same purification table that we have. So what is the fold purification of the most effective step? Okay. Any more answers? Okay. Okay, so hence the correct answer is option number two. Okay, so here also when we calculate the fold purification, okay, so in that case we have to check the specific activity of the enzyme at each step. Okay, and fold purification is given by the specific activity of the given step divided by the specific activity of the initial step. Okay, so from the previous question that we can see, the specific activity of the initial step was 50, if I'm not wrong. Wait a minute. Sorry, it would be 200. Okay, so it was 200, right? Now, when we see this, so specific activity of this step is 200, and for the step that was most effective was 15,000. Okay, now if we for fold purification, if we divide 15,000 by 200, that means it is 75. Okay, so that means the protein has been purified from the first step to the this particular step 75 folds. Okay, so this is the maximum purification that we can get from this particular steps that we are following. Okay, so the correct answer is option number 2, that is 75. So are there any doubts? Okay, so in this case we are calculating the fold purification. Okay, that means how much folds the protein has been purified, right? So if you have to give a number to the particular step, or that you have to tell what is the percentage of the protein that has been purified from a given particular solution, so in that case we calculate the fold purification. And if you have to tell what is the most effective step, or which step leads to the maximum purification. Okay, so in that case the last question comes into play, although the thing that we are getting is similar, because the fold and the effective step is one and the like similar aspect, but the final product that we are asking is different. So is it fine? Okay, so let's move on to the next question. So during amino acid synthesis, we have seen the juxtaposition of two cysteine residues forms a covalent bond, which stabilizes the protein structure. This bond must be broken during sequencing for the undisturbed adduct based cleavage to continue sequencing. Which of the following is or are true regarding this process? So these are the statements given. You have to check which is the correct one and mark the correct ones basically. Okay, so please read them and let me know the answer. More than one option can be correct in this case. Okay. B, C, E, and H. Okay. This is B, this is C, this is E, and this is H. Okay. Any more answers? B, F, H. Okay. F is also included. Okay. Any more answers? Okay. Okay. So let's see the answer. So the correct option are B, C, F, and H. Okay. So we'll see one by one. The molecule with the covalent bond is called cysteine. No. The cysteine is a amino acid. Okay, the single amino acid that have the sulfur group. Okay, so the R molecule present in cysteine is SH only. Okay, so this helps in forming the sulfide bond with the other cysteine molecule. Okay, so this is incorrect. Then the bond that to be broken is a disulfide bond. Yes, so this forms a disulfide bond with the cysteine that is present in a peptide backbone. Okay, and this bond that is formed is a covalent bond and it is called as a disulfide bond. Okay, so the second option that this bond is called as a disulfide bond is correct. Now the molecule with the covalent bond is called cysteine. So when this molecule is formed that have a disulfide bond with them, so these two cysteine molecules forming the disulfide bond is called as a cysteine. That is a single molecule. Okay, because it is attached with a disulfide linkage. So yes, this option is also correct. The bond to be broken is a peroxide bond. No, this is incorrect. Then the bond is broken by adding performic acid, which gives cysteine residues. No, this is false. When we react with the performic acid, it gives cystic acid residues. So the F option is correct. Okay. Now the bond is broken by the addition of DDT, which is further acylated by iodoacetate to give acetylated cysteine residues. No, this is the wrong option. DDT is uh, it's a explosive, right? So it is not used here. It's the DTT, that is dithiotritiol, which is after which it is acylated by iodoacetate to give acetylated cysteine residues. So H option is correct. Okay, so the correct ones are B, C, F, and H. And here is the explanation of the same. So this is a protein molecule that have the cysteine residue. Okay, so this is one cysteine residue and this is the other cysteine residue. The bond that is formed between the two is called as a disulfide bond. During sequencing, what we have to do, we have to break off this bond to get a single amino acid while sequencing. Okay, so first we can do the oxidation by the performic acid. So in that case, what what happens? The bond is broken. This bond is broken, and now the two molecules, the two moieties that are formed are called as cystic acid residues. Okay, and when we reduce it by the dithiotritiol, so this is a structure of the dithiotritiol. Okay, now when we reduce it, then the bond is broken, just like it forms the cysteine residues again, and when it is acylated by iodoacetate, it forms acylated cysteine residues. Okay, the acetyl group is added to the sulfur of the cysteine residues. Okay, so this is how the sulfide bond is broken during sequencing or any other like folding, or during folding reactions also this bond is broken. Okay, so any doubts in this question? Okay, if not, then we'll move on to the next question. So this is the next question. Which of the following are false regarding the chemical synthesis of the proteins? So these are the statements, and let me know the false option. Okay, any answers? A, D, E, G, H. Okay, A, then D, then E, G, and H. Okay. Any more answers? Okay. So the correct answer is A, D, E, and E, G, and H. Okay. So let's see the options. So the chemical synthesis of proteins is used to synthesize long peptide sequences. No, this is false. So the first statement is false because it is used for preparation of the short peptide sequences. Okay. Then it is used to synthesize synthesis of epitopes to activate antibodies for parts of the protein. Yes, this is correct because the whole part is not an epitope of a particular amino acid. Okay, there are different parts of the protein that act as an epitope. So this is correct. Now, used to synthesis of epitopes to activate antibodies for entire proteins. No, this is false. So the A and D are false now. Then E, the amino acid functional group is protected by FMOC, which is attached to the alpha carbon. No, this is also not correct. Then the finished peptide is deprotected by HF, which cleaves the linkage between the peptide and the resin. So yes, this is correct. HF is basically used to cleave the peptide and the resin linkage. Okay. Now the finished peptide is deprotected by HCl, which cleaves the linkage. No, HF is used, so this is also false. The starting molecule is covalently attached to a liquid phase matrix. No, it is attached to a solid phase matrix. Okay, because it need a bead to attach, like it acts as a base for the attachment of the particular peptide. So this is a solid phase matrix rather than a liquid phase matrix. Okay, so the procedure is this. Okay, so here is the FMOC that have the uh, residue, and FMOC is attached to the nitrogen atom of the peptide bond on the alpha carbon chain. Okay, so when it and it is attached to the insoluble, that is solid phase matrix bead. Okay, so once this FMOC is attached to the first amino acid is attached to this particular uh, matrix, then the protecting group is removed. So this FMOC is removed, and this remains only the first amino acid, the mild organic. Now this is the first amino acid. Now FMOC acts with the another amino acid, and that the DCC, dicyclohexyl carbamide, helps in the reaction. Okay, so the after the amino acid is added to this particular thing, then the second amino acid is added after this. So this yellow one is the second amino acid. So this is added, and the uh, dicyclohexyl carbamide is released as a dicyclohexyl urea byproduct. Okay. Now this process continues if we have to add some of the molecules of some of the amino acids after this. Now the complete peptide is released from the solid phase matrix by the addition of HF that cleaves the ester linkage between the peptide and the resin. Okay, so this is the whole procedure of the chemical peptide synthesis that we use. Okay, so any doubts in this question? Okay, if not, then we'll move on to the next question. So now we'll be starting with the enzymes wala part. That is the basic major thing that we have studied in the third week. So which of the following are true regarding the enzymes? So what can be the correct answer? More than one option can be correct in this case also. C and F. Okay, so we have C and F. This is the first answer. Anybody else? C. Okay, let's see the answer. So C and F are the correct answers here. So let's see the statements. So they are the fibrous proteins. No. Fibrous proteins are generally involved in the structure component. Okay. So all the proteins that have a bit of the flexibility in terms of reaction or something, those proteins are globular, not fibrous. Okay. So the first option is correct, incorrect. Collagen is an enzyme? No. It's a structural molecule, structural protein. It supports the cell or the tissue. Okay. They are spherical and folded, yes. Enzymes are spherical, nearly spherical, and these are folded proteins. Okay. So suppose this is a particular enzyme and this is the active site because it tends to react with different active sites. Okay. Different substrates. So it needs to be flexible in nature. Okay. And this active site is formed after folding of the amino acids. So they are spherical and folded. Coenzymes are inorganic molecules? No. Coenzymes are basically organic molecules. Cofactors are inorganic molecules. These are metal ions basically. Okay. An enzyme with incomplete activity is called a holoenzyme. No. The holoenzyme is the combination of the apoenzyme, that is the protein part, and the coenzyme, cofactor, or prosthetic group. Okay, so these three are the additional components of the enzyme that helps in the reaction, that helps in the substrate to product conversion. Okay, so if we combine these two, that forms the holoenzyme, and apoenzyme is only the protein part. So apoenzyme is just the polypeptide chain. Yes. Okay, so the correct options are C and F. Okay, is it fine? Any doubts? Okay. Okay, let's move on to the next question. Which of the following is or are true regarding the coenzymes? So these are the different statements given regarding different coenzymes. Just check what can be the true statements. A, C, D, right? A, C, and D. So the correct answer is A, C, and D, and this will help to understand better. Okay, so these are the different coenzymes that have been found to be involved in the different enzymatic reactions. Okay, so that, like, biocytin or biotin helps in transferring of the CO2. Okay, and dietary precursor in mammals is biotin, right? Now the coenzyme A, it helps in transfer of acyl groups. Then we have coenzyme B12 that helps in transferring of the H atoms and the alkyl groups. FAD transform electrons, lightweight electron transfers electrons and acyl groups. NAD, that is nicotinamide adenine dinucleotide, hydride ions are transferred with the help of NAD. Then pyridoxal phosphate transfer amino group, tetrahydrofolate transfer one carbon groups, okay, and the thymine pyrophosphate transfer aldehydes. So the biotin helps in the transfer of CO2 from one substrate to another. Yes, this is correct. Riboflavin transfers amino groups. Yes, this is also correct. Niacin helps in transfer of hydride ions. No. NAD helps in transfer of the hydride ions. Niacin is the source of NAD. Okay, so this is false. Coenzyme A helps in the transfer of acyl groups. Yes, this is correct. So the correct option is option A, C, and D. Okay. So any doubts in this question? Okay, if not, then we'll move on to the next question. Which of the following is or are true? This is also related to enzyme substrate reaction and the transition of substrate to the product. So, what can be the correct answers? Okay, okay. So, the correct answer is G. So, that means none of these statements are correct. Okay, so let's see. Biological reactions mostly occur without enzymes? No. All the reactions that have been happening in a biological setup in our body, those happen with the help of the enzymes only. Okay, enzymes can decrease or increase the rates and change the equilibrium of the reaction. No, it will never change the equilibrium of the reaction. It can increase or decrease the rate or the energies that have been used to conversion of substrate to product, but it will not be able to change the equilibrium of the reaction. Okay, enzymes are non-specific in nature? No, they are very specific in nature. The substrate that is involved in the enzyme is, like, the enzyme decides which substrate to act on. Okay, so they are very specific in nature. There can be only one substrate in any reaction. No, there can be multiple substrates that can be reacting in a particular reaction. So this is also false. There are no only non-covalent interactions involved between the enzyme and the substrate. No, this is also not correct. Okay. Enzymes provide the energy required to cross the activation energy barrier. No. Enzymes basically help them to interact. Okay, so the energy that is being required to cross the activation energy barrier is between the reaction that helps the enzyme to substrate, followed by the formation of the product. Okay, so this is also false. So let's move on to the next question. In general acid base catalysis, what can be the correct answer from the given statements? Okay. Any more answers? Okay. Any more? Okay. So the correct answer is option B, C, and E. Okay. Okay. So let's see the statements. The proton donor or acceptor is H2O. No, not every time. Okay. So this can be any amino acid side chain or the catalyst that can be involved. So this is not the correct statement. Involves the formation of a stable intermediate? Yes, this involves in the formation of a stable intermediate. The transient stage is intermediate is a stable charge structure. Yes. So what happens whenever a substrate comes and it interacts with the enzyme molecule? So there is a charged intermediate that is formed. Okay, so this is the best example of the acid base catalysis in which a charged intermediate is formed. That next step is the conversion of this charged intermediate to the product. Okay, now moving on to the option D, that is involves the formation of the hydronium ion, not necessarily. Okay, directly H plus ion can either be donated or accepted from the given substrate or product substrate molecule. Okay, so hydronium ion formation is not necessarily needed. Now when the catalyst provides a proton, it is called as acid catalysis. Yes, so whenever the catalyst that is being used is providing a proton, so in that case it is called as an acid catalysis. not the base catalysis. Okay, so the correct answer is option B, C, and E. Okay, so any doubts in this particular question? Okay, so acetyl is basically CH3 COO group. Okay, and acyl is uh, I think the same group that is being formed. That is acetyl group. Okay, because in acyl group, what the main difference is, this CH3 is being replaced by R. Okay, so whenever we are this R can be any carbon chain long. Okay, so for example, if we have something called as uh, like many fatty acids are there that is involved in formation of the whole fatty acid chain or the formation of lipids. So in that case, this R group or acyl group is transferred to the other form. Okay, so this when this CH3 group is shifted to the R group that have many carbon chains. So in that case, it is acyl group, and this is the acetyl group. Okay, shall we move forward? Okay. So this is a biological reaction that is being given. Okay. So that is enzyme catalysis basically. So which of the following is or are true with the with the reaction that is being given above? Okay. So we have got the first answer. Anybody else? Okay. So the correct answer is option number D. Okay, that means none of the above statements are correct. Okay. So the Newth pathway for the reaction has a greater activation energy compared to the uncatalyzed reaction. No, because here the activation energy is being uh, given down by the X molecule, that is basically the enzyme molecule. Okay, the hydrolysis reaction does not readily takes place. Uh, no, it can readily takes place between the B and the H2O because hydrolysis is the removal of a water molecule, so it can readily takes place. The amino acid side chains and cofactors can form non-covalent bonds with the substrate. Yes, but here, wait a minute. The amino acid side chains and cofactors can form non-covalent bonds with the substrate. No, because it has to form some bond so that here, like the substrate is attached to the enzyme. So in this case, it would be a covalent bond with the substrate as compared to the non-covalent bond that we see in other reactions. Okay, so this is also not correct. So the correct answer is option number D. Okay, so let's move on to the next question. Which of the following is or are false regarding the metal ion catalysis? So what can be the correct answer? Okay. Any more answers? A and D. Okay. Any more answers? Okay. So let's see the answer. So the correct answer is A, C, and D. So these statements are false basically. Okay. So in case of metal ion catalysis, the metal ions in these reaction exist only in a single oxidation state? No. Okay. Why? Because if we take example of any of the catalysis of the metal ions, okay. So in that case, it can exist in at least two oxidation states because generally it is involved in the action of the enzyme reaction. Okay. So for example, hemoglobin have iron in Fe2 plus and Fe3 plus state. Okay, Fe2 plus helps in basically binding of the oxygen. So when it binds, it snatches the electron and it converts into Fe3 plus state. So this is both oxidation states are present in the metal ions. Okay. Now metal ions orient the substrate for a given reaction. Yes, this is correct because this helps in orientation of the substrate molecules in the active site. Okay. Then metal ion stabilizes the charge reaction transition state or as the charge on them is lower compared to the amino acid side chains. So metal ions stabilize the charge reaction transition states as the charge on them is lower compared to the amino acid side chains. So no, this is false because metal ions have more charge as compared to the amino acid side chains basically. Okay. Then participate in oxidation reduction reactions by irreversible changes in the oxidation state of the metal ions. No. The it although participates in oxidation reduction reactions, but it is reversible change. Okay, so whenever the work is done, the oxidation state of the metal ion is followed back. So the original oxidation state of the metal ion is preserved every time when the enzyme is in reaction. Okay, so this is also false. So the correct answer of the given question is option A, C, and D. Okay, so any doubts in this question? Okay. If not, then we'll move on to the next question. Which of the following is or are true regarding Michaelis-Menten kinetics? So please read the statements and give the answer. A, C, and F. Okay. Any more answers? Okay, A, E, F, then C also. Okay. Okay, let's see the answer. So the correct answer is option D and option F. Okay. So the rate of the reaction is directly proportional to the substrate concentration. Actually, this is not truly correct because when we calculate the rate of the reaction, it also depends upon the. So this is the equation that we study. Okay, so this can be considered as a constant, but this here that in the denominator it's in the addition sign. So this is not directly proportional to the substrate concentration. Okay, so this option is false. Then the reaction rate reaches Vmax after a long time. So this is not generally correct because Vmax was reached in certain amount of time. Okay, because the enzyme molecule is actively participating and decreasing the activation energy. Okay, the formation of enzyme substrate complex takes a long time, but the conversion of the complex into product occurs quickly. So no, this is false because the enzyme substrate complex takes like occurs quickly, but when this enzyme is like reacted with the substrate and forming the enzyme substrate complex, now the reaction from this to the product formation is a time taking one and it's a complex one. Okay, because the binding the activation on energy of the substrate to the product is very high, so in that case it takes longer time to convert the substrate to the product. Okay, so this D option is correct. The saturation effect is when there is excess enzyme to combine with the substrate. No, but the saturation effect is seen when the enzyme molecules that we have are less. That means when the substrate molecule is more. So in that case, the saturation effect is seen. That means the F option. Okay. And when Km is equal to S, then V naught is equal to V max by 3. No, this is false because when is equal to S, then that in that case V naught is equal to V max by 2. That means half maximum velocity. Okay, so the correct answer is option number D and option number F. Okay, so is the explanation clear? Okay, so let's move on to the next question. So this is a line viewer burp plot that is given, and the slope of the this particular line is zero point 0.5. The area of the triangle, this area is given. Okay, and this is 0.125. So which of the following statements is true? option B and option C. Okay, any more answers? Okay, the Km value is minus 1.45. Okay, okay. So the correct answer is option number A. Okay, so let's see the explanation. So this is a Line-Weaver-Burke plot. Okay, so the y-axis is given by the 1 upon V naught, that is the reaction rate, and the x-axis is given by the 1 upon substrate concentration. And this point is the value of minus 1 by km, and this is the value of the 1 by Vmax, and the slope of this slope of this line is km by Vmax. So here the slope is given as zero point 0.5. So zero point 0.5 is equal to Km by Vmax. And this area is 0.125. Okay. So 0.125 is equal to half. So this is a angle triangle. So the area of a right angle triangle is half into base into height. So this is the base and this is the height. Right. So the base is equal to 1 by Km and the height is equal to 1 by Vmax. Okay, so from this particular equation we can get Km is equal to zero point 0.5 Vmax. So if we put this particular thing here, then in that case it would be 1 upon 2 minus 1 upon zero point 0.5 Vmax into 1 by Vmax. Okay, so the Vmax that is coming here is 2 into zero point 0.5 into 0.125, that too minus, and this is Vmax square. Okay, so Vmax, that is square root of a negative number, so that means that it is a, okay, sorry, so here we'll not put minus sign here because the area is always a positive one, right? And here only the area is given, that means we'll consider only the values. So Vmax here will be this is 10, so this will be going off, so this would come on to be 1.25. Okay, so the Vmax value would be, so the square root of 125 is basically 15, I guess. That is around 11.18. Okay, so if we now multiply it with zero point 0.5, this comes out to be, km comes out to be 5.59. Oh wait, so this comes out to be 1.118 Vmax. Okay, and if we multiply this thing with zero point 0.5, then in that case it comes out to be 0.59. Okay, so the Vmax value is near to 1.41. So option number C is correct. Okay, so this is the wrong option. Option C would be the correct answer. Okay, So is it clear? Sorry, the option was written wrong here. It would be option number C. So is it fine? Yes, you can ask.
While calculating Vmax, it should be, I think, 1 upon zero point 0.5 into 2 into 0.125, which you said in the, like you only multiplied each three number, but it should be 1 by zero point 0.5 into 2 into 0.125
Okay,
in order to calculate the Vmax.
okay, okay. So because uh, here I'm substituting Km, right? So from the slope we can get zero point 0.5 is equal to Km by Vmax, right? So I have substituted this particular value in a denominator because here the slope, that is OA, okay, this is 1 by Km. Okay, so I have substituted this Km value as zero point 0.5 Vmax. So that in that case, it would be going into the numerator. That's why it will be multiplied. But this would be in denominator, I guess.
But sir, it will be 0.125 into 2 into 0.5 V max square equals 1. And when
Yes, yes,
we
yes.
calculating V max,
That's
it
what
will
I
be
got
1
it.
upon,
So Vmax square will be equal to 1 divided by 0.125
yes.
into 2 into zero point 0.5. Okay, I got it. I got it. So that would come out to be, wait a minute, 10 divided by 1.18. So this would be, okay, so this would cancel out. So this gives us out to be 1000 divided by 125, right? So if we calculate this, this comes out to be 8. And if we do square root of 8, this is 2.42. Okay, so 2.42, and if we multiply this with zero point 0.5, this will be 1.4. Fine. Fine, fine, fine. Sorry from my side. This is a correct explanation. Okay. So I'll do the explanation again. Okay. So I'm just erasing it. Okay. Yeah. Thank you for letting me know. Okay. So the slope that we have is zero point 0.5, that is equal to Km by Vmax. Okay. Now this is the area of the right hand a perpendicular triangle. Okay, so this is half into 1 by km into 1 by Vmax. So now if we substitute like this, so from here we got km is equal to zero point 0.5 into Vmax. Right. Now if we see Vmax square is equal to 1 upon 0.125 into 2 into zero point 0.5. Right. Now if we get Vmax is equal to 1000 by 125, that equals 8 Vmax square basically. And if we see, this Vmax is equal to 2.82. After square rooting this 8, we will get 2.82. And if we substitute this back here, we'll get Km is equal to 1.41. That is 2.82 into zero point 0.5. Okay. So yes, the correct answer would be A only, not C. So is it fine? Sorry, I just miscalculated the things. So is the explanation clear now? Okay. So let's move on to the next question. Enzyme A functions at a rate of 30 micromole per minute when the substrate concentration is 0.001 molar. We know Km is equal to 15 into 10 to the power minus 4 molar and the total enzyme concentration is 2.4 micromolar. How long does enzyme A take to act on one molecule of the substrate? So what can be the correct answer? Okay, so we got the first answer. Anybody else? Okay, let's calculate. Fine. So the correct answer is option number C. So what is given in the question basically? So this is 330 micromole per minute. That is the rate of the reaction. So this is V naught that has been given. Okay. The substrate concentration is 0.001 molar, that is 10 to the power minus 3 molar. Km of the particular substrate is 15 into 10 to the power minus 4 molar. And the total enzyme concentration that we have is 2.4 micromolar. So this is the given things that I am writing just now. Okay. Now, in order to check what is the enzyme time that enzyme A takes to act on one molecule, that means it is asking for the turnover number. Okay, so the turnover number is given by 1 by k cat. Fine. So now for this, we will be needing Vmax first. Then we'll calculate the k cat, and then we'll calculate the turnover number. Okay. So for the Vmax, we'll directly apply Michaelis-Menten equation. So that states Vmax into S divided by Km plus S. Okay. So V naught is 30 micromole per minute. Vmax we have to find out. Substrate concentration is 10 to the power 3 minus 3 molar. Km is 1.5 into 10 to the power minus 3 molar, and substrate concentration is again 10 to the power minus 3 molar. Okay, so I have written Km again as 1.5 into 10 to the power minus 3 molar. So this is one and the same thing as read this. Okay, just the way of representation is different. So now from here, the V max comes out to be 75 micromole per minute. Okay, you can calculate this because Vmax comes out to be 75 micromole per minute. Now, what is the formula for the kcat? It's Vmax by the total enzyme concentration. Okay, so Vmax from here we have got as 75 micromole per minute, and the ET, that is the total enzyme concentration, was 2.4 micromole. Okay, so this can be cut, and this is around 75 by 2.4 per minute. So now if we have to find out the turnover number, so we'll just reciprocal this will be in minutes. So now we have to take the enzyme in seconds, so this would come around into 60 seconds, so this would come around 1.92 seconds. So the correct answer is option number C. Okay, is it fine, the explanation? Okay, so shall we move forward to the next question? Okay, so this is the next question. And so we have a 10 amino acid peptide that was cleaved with trypsin. The following fragments were obtained, and these are in the N to C direction. But that means this alanine is the N terminal, free N terminal, and lysine has the free C terminal. Okay, so this is the first fragment, second fragment, third fragment after treating a 10 amino acid long peptide with a trypsin molecule. So this reaction of the intact peptide with fluorodinitrobenzene followed by acid hydrolysis creates a derivative of isoleucine. Which of the following represents the fragments that would be generated when this peptide is treated with pepsin? Okay, so what can be the correct answer? Okay, this is A. Okay, any more answers? Okay, so the correct answer is option number D. Okay, so so first, the reaction of the intact peptide with fluorodinitrobenzene followed by acid hydrolysis creates a derivative of isoleucine. Okay, so that means the N terminal amino acid that we have is isoleucine. Okay, so generating the full peptide from this, we have isoleucine followed by the arginine followed by either this or this. So these two can be the next amino acid sequence that will in this. Okay, so after this, the pepsin treatment was given to the peptide. Okay, so pepsin basically cleaves after tyrosine, tryptophan, phenylalanine, or leucine. Okay, so out of these four, we have only tyrosine in the question here. Fine. So as it cleaves at the C terminal after the C terminal of these particular amino acids. So that means they are freeing the C terminal of these amino acids. So in that case, if we consider the options, so we have first option as isoleucine and arginine, then tyrosine, valine, proline, glutamate, etc. etc. So in this case, because it is cleaving at the N terminal, this would not be the correct answer. Okay. Then isoleucine, arginine, alanine, methionine, histidine, and then tyrosine C terminal followed by valine, proline, and glutamate. So yes, this can be the correct answer. Then, because in this case alanine is the N-terminal amino acid, so this is already false because isoleucine is the N-terminal amino acid as given by the first statement. Then isoleucine, arginine, alanine, methionine, histidine, and lysine, followed by tyrosine, valine, proline, and glutamate. No, this is also not the correct answer because this is cleaved by the N terminal. Okay, and pepsin cleaves at the C terminal of the amino acid. So the correct answer is option number B, not D. Okay, is the explanation correct? So these are the different enzymes that are basically used for protein sequencing, and these are the amino acids that are basically cleaved by these particular proteins. So trypsin cleaves after lysine and arginine. Then the submaxillus protease cleaves after arginine only specifically. Then chymotrypsin cleaves after phenylalanine, tryptophan, and tyrosine. And Staphylococcus aureus V8 protease cleaves after aspartate and glutamate. Okay. So likewise, pepsin cleaves after phenylalanine, tryptophan, and tyrosine, and leucine also. Okay. And endoproteinase lyse C cleaves after lysine, and cyanogen bromide cleaves after methionine. Okay. Okay. Let's move on to the next question. So this is the same question, just a different part. Which amino acid would be released when the peptide is treated with a combination of endoproteinase lyse C and chymotrypsin? So what can be the correct answer? Any answers? Okay, so here the correct answer is option C, that is tyrosine. So from the last question, as we can see, the correct sequence would be, this would be the first peptide near to the N terminal. This would be the second peptide, and this would be the third peptide. Okay. So when it reacts with endoproteinase lyse C, that means it cleaves after lysine residue and the C terminal of the lysine residue. Okay, so this would cleave after lysine residue, and after this we cleave this with chymotrypsin. So chymotrypsin cleaves after tyrosine, phenylalanine, or tryptophan N terminal residue. Sorry, C terminal residue. Okay, so this fragment is formed. Now the other fragment that would be formed is tyrosine. So the cleavage of chymotrypsin will be after tyrosine. So this fragment will be released. So the only amino acid that we'll be getting is tyrosine from the given molecule. So is the explanation clear? Okay, then I'll explain again. So, from the last question that we have seen, the sequence of peptide will be, this is the first peptide, followed by histidine, lysine, then tyrosine, then valine, proline, and glutamate. Okay, so this was the correct sequence of the peptide that we had from the last question. Now, in this case, we'll be first treating it with endoproteinase lyse C that cleaves after the C terminal of lysine. Okay, and then we'll treat it with chymotrypsin. So, chymotrypsin cleaves after tyrosine, phenylalanine, and tryptophan C terminal. Okay, so it would cleave after tyrosine. So, the only amino acid that will be formed from this particular cleavage of two enzymes, that will be tyrosine. Okay, the rest, this peptide, this would be the N terminal peptide, and this would be the C terminal peptide that will be formed. So now the explanation is clear. Hmm. Okay, so let's move on to the next question. The other question from the same part. So, which of the following would break only one peptide bond in the intact peptide? So, you know the sequence of the peptides from the last two questions. So, now what will be the answer for this particular question? Okay. Any more answers? Okay. So here the correct answer is option number A, C, and D. Okay. So how they can break one peptide bond? Cyanogen bromide breaks the peptide bond after methionine. Okay. So here is the methionine, and here only one peptide bond will be broken. So this is the correct answer. Then N protease breaks the peptide bond from the N terminal of the aspartate. So because aspartate is not present in the peptide, this would be of no use. Now V8 protease V8 protease basically cleaves after the C terminal of glutamate or aspartate. Okay, so here we have one glutamate molecule. So this can be used for cleaving the peptide. Then we have chymotrypsin. So this cleaves at the C terminal position of tyrosine, tryptophan, and phenylalanine. So here we have only one tyrosine. So only one peptide bond will be broken. Okay. So here the correct answer would be A, C, and D. So any doubts in this particular question? Yes.
Sir, in case of V8 protease, it will be going to cleave in the C terminal of either aspartate or glutamate.
Yes.
Since we know that the sequence contain one glutamate at the end,
Hmm.
so if it is cutting at the C terminal, it means there is no cut.
Yeah,
The
actually, I
entire
also had the same question, but as per the question bank that I have, so this is the correct answer. So I also like, and I'm a bit confused about this particular option about the V8 protease and how it can be correct. So that's why, like, I marked it correct because the glutamate molecule is present at the end. But I am also not sure about the option number C. But option number A and D are sure correct. Yes. For the this particular thing, you have to remember the enzymes and where they cleave, because based on that only these particular questions can be answered. And this is basically the sequencing wala part from the protein structure. So in case of sequencing, we use different enzymes to cleave and form the fragments, and after like aligning those fragments we get a full protein structure. So it's better to like remember the proteins that cleave at which particular position of the amino acids. Okay, is it fine? Shall we move forward? Okay. So, based on their functions, what are the first digits of the EC numbers of trypsin and hemoglobin, respectively? What can be the correct answer? Okay, we have got the first answer. Anybody else? Okay, so the correct answer is option number C. That means 3 and does not apply. So EC is the enzyme commission number that is given to the particular enzymes. Okay, so as hemoglobin is not an enzyme, it's a transport molecule. So this does not apply to hemoglobin. For trypsin, if we see, so as we see, enzyme commission number is a four-digit number that are separated by dots. Okay, so the first number gives the main class of the enzymes. Okay, so there are six classes, that is oxidoreductase, transferase, hydrolase, lyase, isomerase, ligases, and the last one that has been recently added is translocases. Okay, So this would be seventh. So total 7. Now the second number that we give is based upon the substrate or the bond that is being broken. Okay. The third one is given based on the subclass of the enzyme, and the last one is given to specifically to particular enzyme. So this is the differentiating value, differentiating number. So for the trypsin, because it belongs to the class of hydrolases, the number that is given is 3. Okay. So this would be the correct option, that is option number C. Okay. So anyone, any doubts? Okay. If not, then we'll move on to the next question. What catalytic mechanism is being followed here? So there can be more than one option that can be correct. C and B. Okay. And A also. Okay. Anybody else? Okay. How can it be metal ion catalysis? Because I cannot see any metal ion that is being involved in this particular reaction. So I don't think metal ion catalysis would be the correct answer. Okay, so let's see the answer. So the correct answer is option number B and C. That means that is covalent catalysis and acid base catalysis. So how? As you can see in the image, this particular group has is belongs to imidazole, basically histidine. Okay, so this H is being transferred to the SH group. Okay, and this H, because it's involving H, that means it is acid base catalysis. And as it is being transferred from this H to the other molecule, that means the covalent bond will be formed and broken. Okay, so that's why it is given as an example of the covalent catalysis also. Okay, is the explanation fine? It is not an example of the metal ion catalysis because here no metal ion is involved here. So this is not an example of the metal ion catalysis. Okay, let's move on to the next question. Which of the following are true for the enzymes? Okay, so A and C. Okay, any more answers? Okay, so the correct answer is option number C only. So Km is always a measure of substrate affinity? No. Okay, why? Because it has two cases. Okay, as we can see in the Michaelis-Menten equation, when Km is equal to K2 by K1, so in that case, it denotes the affinity of the substrate. Fine. But when Km is equal to, when K minus 1 is very, very greater than K2, in that case, it is this. So in this case, the Km denotes the reaction, sorry, the binding energy of the reaction and how the reaction will move forward. Okay, so every time it is not the affinity towards the substrate. The free energy of the enzyme substrate complex is lower than the free energy of the substrate during enzyme catalysis. Okay, no, the other way around is possible. So, because the enzyme substrate complex has free energy that is higher than the only the substrate because it is being converted to the product molecule. So that's why this is a false option. Then zinc ions increases the activity of the alcohol dehydrogenase. Yes, because zinc ions are involved as a cofactors of the alcohol dehydrogenase. A cell can function if the activation energies of all the reactions are extremely low. No, because in that case, enzymes will be not required because the activation energy is so low. That means the substrate can directly be converted to the product molecules. Okay, so the correct answer is option number C. Okay, so that's all for today's class. If you guys have any doubt in this particular presentation, you can ask. Yes.
Sir, are these videos being recorded because I missed first two live classes?
Yeah, no issues. It is being uploaded in YouTube. You can check your NPTEL page. There, the video link will be given.
Or like they will give us via mail, or because we need to type something, otherwise we won't be able to see in the YouTube.
Oh, okay. I'll check and let you know because in my knowledge, what we have seen is there would be some link of the drive that will be given. So in that case, the drive we in the drive you have a sheet that gives you the link of the videos.
Okay, as per my knowledge, it's not there, but I will check again.
Yeah, you check. Otherwise, you can ask in the discussion forum also.
Okay, okay, okay. Thank you,
Okay.
sir.
Okay. If not, then we'll close the session for today. Okay. Thank you guys for joining, and let's meet next week. Okay. Thank you.